my science project, I'll be explaining the cell cycle and its phases. The cell cycle consists of two main phases, interphase and mitosis. Interphase is where the cell does its job as normal and prepares for mitosis. Interphase has three subphases, gap 1, synthesis, and gap 2. Gap 1 is where the cell grows and does its job. Synthesis is where the DNA replication occurs to prepare for mitosis. Gap 2 is where the cell adds on extra volume to prepare for dividing. There is also a G0 phase where the cell just does its job and does not prepare for mitosis in any way. Cells that don't divide spend most of their time in G0, such as neurons or white blood cells. Before the cell moves on from any phase, it has to pass what we call a checkpoint. Checkpoints are there so that if the cell is unhealthy, it doesn't replicate bad DNA. If the cell has bad DNA or doesn't function properly, it will sense that and never divide. Tumors can form if the cell is unhealthy but still replicates. After interphase comes mitosis. Mitosis is the cell's process to divide and replicate its DNA, and it has 5 subphases. The first is prophase, where chromosomes are formed in the nucleus, the nuclear wall breaks down, and centrioles create spindle fibers, which attach to the centromere of each chromosome. But before we can go any further, we need to take a look at DNA's structure. DNA is a double helix, but it wraps very tightly around proteins called histones to form chromatin. Chromatin is the natural state of DNA in the nucleus, and it is very messy. To make DNA easy to replicate, it forms into neat chromosomes, which are made up of 2 sister chromatids which are identical and held together at the centromere of each chromosome. The centromere is just a special protein. Now back to the second phase of mitosis. The second phase is called metaphase, where the chromosomes line up in the middle of the cell because of the spindle fibers attached to them. In the third phase, anaphase, the spindle fibers pull the chromosomes apart at the centromere, so that each side of the cell has its own set of identical chromatids. In the fourth phase, telophase, the spindle fibers dissolve, nuclear membranes reform around each set of chromatids, and the chromatids uncoil back into chromatin. Now that the cell has 2 sets of DNA, we just have to divide it. Cytokinesis is the process by which the cell divides the cytoplasm. It overlaps with mitosis, so we just consider it the 5th phase. In animal cells, the cytoplasm divides along the cleavage furrow and splits the cell in 2 during cytokinesis. During cytokinesis in plant cells, vesicles come to the middle of the cell and form the cell plate, which is like the cell wall, and the cell splits apart. Then, the 2 brand new daughter cells of the parent cell start this process all over again. Thank you for watching, and please give me a 100.